Hi everyone, we are on to lecture 13.3, the third mini lecture out of chapter 13. In this one we're going to be going into stage 2 of the specific immunity, the third line of immunity, and we're going to be looking at antigens. So this is a list of the learning outcomes that I would expect for this lecture. We're going to start with a presentation of antigens, or the entrance of antigens. So as I said, that is also synonymous with the word immunogen. And these are any substances that could elicit or cause an immune response. However, they have to meet certain requirements. So they have to be foreign enough to be recognized as a problem. Their shape and size is important, and also the accessibility, if our immune system has access to these particular antigens. And they must not be a normal constituent of the body, otherwise that would be an incorrect reaction. So some examples that you might see as immunogens, would be whole microbes or their parts, so bacteria or viruses, either the whole microbe or maybe pieces of it. Also, any cells or substances that came from perhaps other humans, animals, plants, or maybe some sort of a toxin, so various molecules. And most antigens, though, will fall into one of the following categories. They'll either be proteins and or polypeptides. They might be lipoproteins, which would be cell membrane proteins. Glycoproteins are one, too, that we might see, and also nucleoproteins, so DNA mixed with proteins, so somehow complex to proteins. Polysaccharides and then lipopolysaccharides are different antigens we might see. So one of the things that we have to keep in mind is that the size and shape does matter when we look at antigens. First of all, they have to be large enough, really, that they catch the attention of the surveillance cell. So in this case, we're talking about macromolecules that are at least 100,000 Daltons. And that's a size that it's big enough that our surveillance cells will notice them. However, it's not just size that matters. So if you have a very large sized molecule, but it is all similar structures, so it's just repetitive subunits, generally it will not be immunogenic. They would need something that has some kind of complexity to the molecule. So some good immunogens that we might see are proteins that have kind of interesting epitopes that we might be able to recognize, or maybe foreign or human animal cells, or animal cells, microbial cells or viruses. We can see some bacteria, some viruses, different things, or plant molecules. What is not a good immunogen would be maybe a trisaccharide, so multiple subunits of a particular carbohydrate, or a polypeptide, just a string that we would see making up a polypeptide, so of amino acids. These are not really different enough. Their shape and everything is too uniform to be noticed. So when we're looking at that molecular size and shape, the epitope is that portion of the molecule that the lymphocyte is actually going to respond to. And the epitope is what signals that immune system that the molecule is foreign. So it's that portion that flags it as being foreign. And we can have something called haptens, and you want to think about these as very small epitopes that are too small to be recognized. And so in this case, they would not elicit any kind of immune response unless it's linked to a larger carrier. And so when it is in combination with a larger carrier molecule, it can then serve as the epitope. So the larger carrier molecule in conjunction with it is that actual antigen, but it's that hapten, that small portion of foreign, that would be considered the epitope of that. So some things that we would see that would be too small on their own, but hopefully can combine with larger carrier molecules. So perhaps drugs, some metals, and some chemicals that we might see are too small on their own. But once they've been combined with carrier molecules, perhaps a serum protein, they would be recognized. And they would then be considered that epitope of the antigen. So in this case, you can see this is just a poor immunogen, it's very small and it's a unique, it's a repetitive subunits. But if we add it to a carrier molecule that makes it seem larger, and it is then unique as that epitope on it, that's what would actually cause a reaction. So with that, we want to think also about alloantigens. And these are cell surface markers or molecules that can occur on members of the same species, but not in others. So this would be something that we would see with blood groups. So you might have heard that you're A or B, AB or O. And it's that within an individual species, we have some members that have different blood groups or different major histocompatibility profile. And so this is where we would start looking at incompatibilities. If you were somebody who had an alloantigen that was different from your neighbor, you would see that there could be a problem with blood transfusion or organ grafting because it would be actually recognized as foreign. So when we start looking at antigens, we also have something called superantigens. And these are ones that are very potent stimuli. So they have a higher stimulation rate than you would normally have with a regular antigen. Some things that would fall into this category would be bacterial toxins. And they are very potent at stimulating the T cells. And they can activate it at a rate of 100 times what would be an ordinary signal. This can cause a real problem because if we get too high of a reaction, what we can have is an overwhelming release of those cytokines, and we can actually cause cell death by having too much of these cytokines being produced. And this is what we would see with toxic shock syndrome and some autoimmune diseases where we have these superantigens that provoke too strong of a reaction. And then one more vocabulary word, allergens are antigens that provoke this allergic reaction. So they provoke an immune reaction, even though that antigen is not really part of something that's harmful. So in this way, it's kind of faking out our immune system in a way that our immune system doesn't recognize that it's harmless, and it will actually provoke this immune or allergic reaction. So how do these antigens get presented? We talked about the fact that they're presented. They need to be presented for a T cell to recognize them. So in most immune reactions, that antigen is going to be presented to lymphocytes by an antigen presenting cell. These are shortened to APCs. Some that you would see those with are macrophages, B cells, and dendritic cells. And these have the ability to break down a particular antigen or a particular molecule and present that antigen on the MHC receptor on the cell. And so in this case, that antigen will be bound to the MHC receptor and move to the surface, and that way T lymphocytes can recognize it as it's presented to them. So this is showing how that might be done. So we have these microbes, bacteria coming in. We have an antigen presenting cell. In this case, it's a dendritic cell. It comes in, it's broken down, and it says, oh, this little antigen is going to be a great one for T cells to recognize. We'll place it on the CH2 receptors, that MHC2 receptor. And what then can happen is that this will then bond with that T cell. So what you get is a T helper cell. It has its CD4, which is recognizing the self in the MHC2, but it also has its receptor, the T cell receptor, that is there to recognize that antigen. And so it's that combination of recognizing self and recognizing antigen that's important in recognizing that it needs to do something. At this point too, once that identification has occurred, then the APC will secrete interleukin 1, and in this case interleukin 1, when it is secreted and the T helper cell is affected by it, it will become activated and can secrete interleukin 2, which is going to help with T helper cells and cytotoxic B cells, and it can also activate the B cell system. So this is all after that antigen presentation to T cells, what occurs right with that first cell that recognizes the antigen. So most antigens are going to be presented first to T cells, and that's even though they will eventually activate both the T and B cell systems, because remember that antigen is still what's going to bind to the immunoglobulin on the B cells. So T cell antigens, these independent antigens, can trigger B cells directly without the APCs or T helper cells. And that would be ones that maybe don't bond a T helper cell, or they haven't gone through the antigen presenting, but they've actually gone directly to the B cell and actually triggered that. And we'll look at that when we talk about B cells and how they get triggered. And at the end, this is the checking in. Please make sure that you recognize the vocabulary and then also the different processes that we went over for this particular lecture. Thanks.